Hello and welcome to Gold Standard Diagnostics webinar session. Today we are going to speak about recent updates on allergen legislation, and we will present your new innovative food allergen detection methods. I am Gábor Kóth, Sales Director for Europe, Middle East and Africa. I will be hosting today's session together with my colleague Cristina Romero, who is our expert and product manager for allergen management. During the webinar, you may have questions. Please feel free to enter them into the chat box as you see it in the screen right now. We will do our best to answer all of your questions after the presentation, when we will have a question and answer part. In case you have questions later on or after the presentation, please feel free to contact us at the email address at contact@eu.goldstandarddiagnostics.com. Now we can go to the next. Yes, thanks. Okay, so before we go into the details, let me quickly introduce you Gold Standard Diagnostics briefly. We are a fast-growing global provider of diagnostic test kits, lab consumables, and instruments, supporting rapid testing in the food, feed, environmental, biopharma, animal health, and clinical markets. Our company has three divisions. The industrial solution is where you find the food, feed, and environmental safety testing tools. The animal health solution provides diagnostic test kits for veterinary diagnostics, both for companion and farm animals. And finally, our clinical diagnostic division supports the IVD, human diagnostic markets. At Gold Standard Diagnostics Industrial Solution, we provide tools for testing at all the steps of the value chain, from the field until the finished products, starting from GMO testing, mycotoxin, environmental and water analysis, pathogen detection, and allergen management as well. And if you go to the next slide, you will see that, of course, allergen management is our topic for today. And you will hear about the regulation part. We will present Vital and PAL concepts. And today we are launching the new generation of rapid detection kits, Powerline, and the multi-3-nut lateral flow devices. We will be speaking about emergent allergens, buckwheat detection with ELISA methods, and we will close the session with question and answers. And with that, let me give the floor to my colleague, Cristina Romero. Hi, Cristina. Welcome.
Hello, Gábor. Thank you very much. And
welcome.
Thank you thank you very much to all of you for joining this webinar. As Gábor has mentioned, we are going to start with reviewing some part of the updates regarding the allergen regulation or the allergen legislation. As you probably already know, the current European regulation for allergens establishes 14 allergens that must be listed or must be labeled in any kind of packaged foodstuffs. So these allergens are cereals containing gluten, crustacean, egg, fish, well, the list that you can see here. Regarding the limits for gluten, which is the only allergen which is currently legislated for limits, according to the Codex standard, the gluten limits are the following. Below 20 ppm, we can label a product as gluten-free. Between 20 and 100 ppm, the product must be labeled as low gluten content. To be honest, from a industry perspective, this is not very practical. So as far as we know that usually they label as gluten-free or containing gluten, because the intermediate solution is quite a little bit risky for the final consumer. And above 100 ppm, the product should be labeled as containing gluten. Also, the Codex established the method for gluten determination as the official one for the ELISA R5 method. If we talk about the rest of the allergens, there is no regulated limits for all of them. But it exists what is called the VITAL program, created by the Allergen Bureau. It's a voluntary incidental trace allergen labeling, and this is a risk assessment process to assist food producers in presenting the allergen advice in a very consistently way for allergic consumers. So it's a support for the manufacturers. This VITAL program establish or define what is called the reference dose. Reference dose are the milligram of protein level below which only the most sensitive of individuals in the allergic population are likely to experience an adverse reaction. So it's like a threshold established based on some clinical research. The VITAL program is not mandatory. It's not a legislation. It's a voluntary decision of the company. But it's true that as there is no regulated limits for the rest of the allergen, most of the companies right now tends to follow this kind of VITAL threshold to establish their internal quality assessment for allergens. This program is not applicable to ingredients, but to cross-contamination. I mean, it is not for when the foodstuff is containing a specific allergen. It's when there is a possibility of cross-contamination. The idea is to avoid the indiscriminate use of the PAL, the Precautionary Allergen Labelling, what is called the may contain. So the may contain sometimes refers to products that probably they do not contain this kind of allergen, that they may have some kind of cross-contamination. It means a quite strict limitation from some of the consumers that probably they can even finally intake this kind of product. So with this VITAL threshold, it is perfectly established in which moment or what action can be taken, so we can establish or we can label the product as may contain or not contain a specific allergen. Recently this year, in August, it was launched the last version of this VITAL program, the version 4.0. So the main difference between this version and the last one is the ones that I'm going to explain now. So first of all, I want to explain that the reference doses that we have just mentioned, they are based on the ED values. The ED is the eliciting doses. The eliciting doses, they are calculated in an experimental or clinical research. So currently in the current version, these ED doses have been changed from the 0.1 to the 0.5, right? So it means that the reference doses have changed from the last version to the new one. And then if the reference dose changes, it means that also the action level has also changed. So the moment that we need to take some action when we need to indicate a PAL statement or not, the values right now are different from the previous one. This is one of the main changes in the current version of the VITAL program. Also, they have included the mollusk as an allergen because it has a very regional significance in Australia and New Zealand, but it was not included in the previous one. The third change is that the Canadian legislation is also in the VITAL online. It means that all the food market inside Canada, within Canada, now they are able to follow this VITAL assessment too. And the last one, but not least important, is that so far, Netherlands was the only one which has already included the ED 0.5 in mustard and lupin, but not the rest of Europe. But now that the VITAL has also included, it means that we are now all harmonized. So all European countries and also Netherlands can use the VITAL online for all kind of market between them. So it really is quite positive because now we all follow the same rules or the same values, to say so. So this is currently the situation with allergen. So the real regulation is the European one to label, but right now the VITAL is really considering in many industries when they establish this milligram of protein and they establish the intake value to calculate the ppm or the limit of ppm that the kit should have so they can use it or they can launch a product with a specific limit of detection or allergen content. So which is the solution that we can offer from GSD? So as a general overview, we can cover all the allergen solution in the different fields. So we have a SensiSpec portfolio, it's ELISA portfolio, including the gluten R5 reference method. We have the SensiStrip portfolio that we will explain later, and the DNA allergen PCR kit. So the three key methods for detecting allergens. We also have some instrument offer, the ELISA reader, the automation with Bolt and Thunderbolt to be able to perform the complete ELISA. We will also see it later more in detail. And of course, the possibility of quantification of the LFTs by using the RapidScan reader. And as complementary product, we also have the swabbing kit, some spiking solution, which is quite unique comparing with other companies. So you can prepare samples with a specific amount of allergen and to use it as an internal control or to check that everything is okay. We also have allergen control material and a specific department for so complicated or complex matrix validation. So let's start with the ELISA product line. These are currently all the ELISA kits we have available. So you can see that we cover all the ELISA kits, all the allergens that are mentioned in the European regulation, excepting the lactose and the sulfites, because these are not proteins and this should be used a different technique to be able to detect them. But for the rest, we cover all of them, even the nuts, because in the European regulation is mentioned nuts, but not the specific nuts. So finally, it's necessary to detect hazelnut, cashew, coconut, macadamia nut, so all pecan nut, all the different nuts that are available in the market. In the case of gluten, which is probably one of the key allergens in the market, we have three different kits. They are what we call the SensiSpec increasing gluten R5, the normal one, which is AOSC validated. The quick one, which is very similar to the previous, but with shorter time of incubation for the ELISA, and also the hydrolyzed gluten R5 kit, which is a specific design for hydrolyzed samples, mainly validated for beer, but also for some other alcoholic beverages and also for baby food, which are typically hydrolyzed samples in the market. So for the AOSC validated kit, we can highlight that the high sensitivity, we are talking about a limit of quantification of 3 ppm of gluten. As you can see, it's quite far below the 20 ppm that we have seen in the Codex Alimentarius according to the legislation. This is a valid kit for the ISO 17025 accreditation for the labs. So there is no problem to using this kit for this validation. And a very important point that the kit does not include toxic reagents. So usually in the case of gluten, previously it was used some beta-mercaptoethanol or some other reagents that may be toxic for the user. And in this case, we are using a completely different extraction buffer, even same effective, but the main advantage is there is not need to use it under fume hood, and of course not bad smell in the laboratory or in the industry. And this extraction buffer is also included in the kit. So as a general idea, we can see that currently we have the biggest product range in the market for ELISA kits with high sensitivity, accuracy, and reproducibility. The performance is quite easy, and we are talking about incubation time of 20 minutes of samples and standards, 20 minutes of conjugate, and 20 minutes of substrate. This is quite convenient for the labs because this way first of all, it's quite easy to remember, and also there are short times, but at least they are long enough so you are able to perform a whole plate if you want, with not working or performing the ELISA in a rush. Also, an important point is that we have a common extraction procedure for most of the kits. It means that the same extract can be used for different kits, which is really reduce the workflow in the lab a lot. not for all the kits, but mainly for most of them. The kits are available in the 48 and 96 wells format. And of course, we participate in many ring trials to check the features of our kit, mainly FAPAs and in all the DLA that are available in the market. We also see now the automation of the kits, which are all of them validated both in the Bolt and the Thunderbolt. So these instruments are the one that I am going to show you now. So we can perform the analysis in a manual way. In that case, we need to use a washer or wash manually. We need to use a reader and then perform all the calculation of the kit. But we also have the option to use a fully automated system. In this case, you have two automated systems, Bolt and Thunderbolt. The main difference between these two instruments is that the Bolt is for one plate, while the Thunderbolt is for two plates. And the main advantage is that in that case, you can put all the reagent and all the samples in the racks, program what you want to test, gluten, almond, peanut, or different, up to 6 different allergens at the same time. And then you let the instrument to work, and it performs everything. So it add the reagents, the washing step, the incubation, and make the final calculation. So finally, you get a full report with all the calculation already done, and with all the plate or even a few strips. You can even let the instrument working over the night, and then see the results during the next day, because the instrument advises you when there is any kind of problem in a specific well, and then you can go on working with them. So this is a very, very convenient solution for big labs, or maybe not so very big, but when you have many samples of gluten or many samples of allergens. These instruments are also valid for other kits like mycotoxin or pathogens with no problem. And now let's go to the strips portfolio, the lateral flow devices. Again, this is the complete list of the kits we have available right now. You can see that they are all validated in food matrices, surfaces sample, and also rinse water. And here you can see that some of them we are marked as new, which is the power line kits and the multi-trinat, which is specifically the new generation of rapid test kits that I want to explain right now. So let's start then with the power line. And to be able to explain the power line's new generation, first I need to explain a little bit about what is the hook effect. The hook effect is something which is related with the lateral flow technique. So it happens when the concentration of an allergen is too high, that the intensity of the test line starts to decrease. This is something that happened with all the lateral flow device strips. So with the LFD power line, we overcome this limitation by including a fill line, indicating if a negative result is really negative or it comes from an excess of allergen in the sample and the consequent hook effect that it provokes. So in the power line, the interpretation of the result is like you can see here. So we have 3 lines. We have the control line, the hook line, and the test line. So these are the 4 possibilities. So in case number 1, we have a line in control and hook, and there is no line in the test. So it means that this is a negative sample. In the example number 2, we have clearly 3 lines. This is a positive sample with positive in the test, hook, and control line. The third example, in this case, we start to see that the hook line disappears, but the test line is still positive. It means that this, of course, is a positive sample, but it's a very high positive because the hook line starts to disappear. And we have the most important case. When the sample is very, very positive, then both hook and test line disappear. So in a normal kit, in this case, you don't see the test line, you could think that it is a negative result. But now, as the hook line has also disappeared, it indicates you that the real concentration is a very high concentration of the sample. Usually, we are talking about samples that contains the allergen as an ingredient. We're talking about 1,000 or yes, 20,000 of ppm. But it's also important to be able to distinguish. The procedure for this kind of strips, not only the power line, but all the strips, is quite simple. You can see here that first we have the sample preparation, which is in fact extraction of the sample. Then we transfer the supernatant into an incubation vial and then place the strip. And after only 5 minutes, then we have the results and we can evaluate the strips. The only difference is with the gluten kit, because in this case we have an additional step because, as you probably know, the proteins from gluten, they need to be extracted in ethanol because they are not soluble in water-based buffers. So it means that after the ethanols, we need to perform an extra dilution step in order the ethanol does not affect the antibodies. It's the only extra step, a dilution one, but the rest is exactly the same. And this, what I've just indicated you, is how to do it in a with a visual interpretation of the result. But here we offer the possibility of obtaining a quantitative result by using the rapid scan reader. So this reader is specifically designed for field and in-process testing. It's applicable not only to allergens, but also to GMO, mycotoxin, animal health, algal toxin, and also glyphosate from GSD. As you can see, this is a very hand-friendly device, not very big, no training is needed, it's very easy to use. We will see now how to use with the strip. It's practical and multitasker with many compatible applications, and it provides fast and quantitative results, which in some cases is quite important. Especially in the case of gluten, because as the regulation indicates 20 ppm, so it's not the same if you have a positive result of 5 ppm that if you have a positive result of 25 or 30 ppm or even more. So in that case, to have a quantification is something that could be quite important. So how can we manage, or how can we deal with the reader? You will see how simple it is. So after we perform the strips, the analysis according to the instruction for use of the kit. So we have not only allergen, the same for the rest of the analysis. Then you have the strip, and you should put the strip in an adapter. There are different adapters depending if it's allergen or mycotoxin or different analyte. And then you insert the stripped adapter in the black hole of the reader. Next steps is just press the red strip button, and then you will see the screen that I show you right here. So now you have to select the kit from the list of already uploaded methods that you have put in the instrument, and you select the kit and the lot. And when you have selected it, then automatically you will see the lot number and the expiry date and the analyte, because it's automatically recognized. You should change the dilution factor, usually is 1, and you can put the name of the sample ID. In this case, I have gluten cake mix as if were a FAPA sample. Then start analysis, and then we can view the results. So these are the 4 options for a power line, but in this case, how do you see it in the rapid scan? So when the sample is negative, you will see below the LOQ. In this case, the example is gluten, and the quantification range in gluten goes from 4 ppm to 80 ppm. So if the sample is negative, you will see below 4 ppm. If the sample is positive within the range, you will see a value, 11 ppm or 12 ppm or whatever it is the value. When the sample is above the quantification range, but not too much, you will see above 80 ppm or whatever is the upper range it is, if it's not gluten, if it's almond or any other allergens. And quite important here with the power line is that in the case you have a very, very positive sample, so in that case, you will see that the test line is almost disappear, or at least is very faint. So in this case, but the hook line has also disappeared. So it means that the sample, as we have explained before, is very, very positive. So here you will see the hook effect has happened, and you will see it directly in the screen. No need of another interpretation. So as a general overview of our census strip line, we can say that we have an easy to use lateral flow format. They give a rapid and accurate on-site test. The kits are provided with all the necessary material, so there is no need to buy any disposable material. You even have the spoons to take the sample, the spatulas to take the samples, the plastic pipettes, and we offer it in 2 different formats, 5 or 20 individual analysis per kit. As you saw before, it is one-step process. So it's only 5 minutes of incubation after destruction, and there is no other step that you need to perform. And only 5 minutes incubation time. It's high sensitivity and specificity. This is thanks to the internal design and the antibody that we used in the different kits. Of course, we offer the possibility of the rapid scan reader with all our kits. The example I have shown here is regarding the power line, but we have the same option for all the allergen kits. And we are currently developing a whole portfolio of power lines kits. Currently, we have available gluten and soy, but we will have at the beginning of next year, we will working already on peanut, milk, and casein. And the idea finally is that we will have a complete new portfolio of power line kits with the hook line. So now let's go to the new multi-trainer kit, which will be available very soon. In fact, the launch will be exactly next week. And the main advantage of this test strip is that it's able to detect 8 different tree nuts in only one strip. So you probably know that tree nut allergy is one of the most common food allergens. It's affecting about 0.5 to 100% of the population. So it's close to the celiac disease, which is really a high percentage. And in this case, sometimes very low amount of tree nuts may cause severe symptoms like anaphylaxis or even different symptoms. In affected people, we all have heard about different news regarding people that have very rare or impressive symptom only taking a little amount of a tree nut. So the sensitive multi-tree nut LFD represents a highly sensitive detection system which is capable to detect residues of almond, Brazil nut, cashew, hazelnut, macadamia, pecan, pistachio, and walnut in rinsed water and swab samples, and everything in the same strip. Now we are going to see how it works. So here, regarding the sample procedure and the testing of the street is exactly the same that we have seen for the power line. So extract the sample and then test in 5 minutes. There is as easy as we have just seen. So what we are going to see here different is the interpretation of the result. So these are the strips for the multi-tree nut, and again we have 3 lines. We have the control line, we have the T1 line, and in T1 is able to detect, is where we can detect almond, Brazil nut, cashew, and pistachio. And we have the T2 line, where we can detect hazelnut, macadamia, pecan, and walnut. So in that case, if we see the example 1, we only see the control line, so the sample is completely negative for these 8 tree nuts. If we see the control line and the T1, it means that the sample contains almond, Brazil nut, cashew, or pistachio. If we only see T1, it means that the sample contains hazelnut, macadamia, pecan, or walnut. And of course, if we see the 2 lines, it means that the sample may contain any of these tree nuts. So, of course, in this case, it's not possible to make a quantification because we are detecting different allergens, different tree nuts in the same line, but it's very, very convenient because if you remember, as I have shown you before, the European regulation indicates nuts. So if you want to label a product as multi-tree nut free or nut free, then you need to perform different ELISA kits or different strip kits. So in this case, with only one test, you are able to discard 8 different tree nuts in only one test. So it's really, really something that makes life easier for the food industry and also for the labs, so as a very quick screening test. And then we are going to see what is called the emerging allergens. So we can explain also the ELISA method for buckwheat detection. So what is called emerging allergens? So, or why this allergen happens in the foodstuffs? So according to the FAO, the changes in consumer dietary preference and innovation in agriculture and food processing can come with the introduction of new hazards and or routine of exposure. So of course, as we are introducing new foods, new process, so it means that currently maybe the 14 allergens which are already listed in the European regulation they are not enough. So we are creating more and more new product that may cause allergic reaction in some sensitive people. So the food business operators, it means the food industry, they have the responsibility to provide safe products for all the consumer, including people with food allergens. So the key to do that is the combination of pre-market safety assessment of alternative proteins, a clear labeling, and post-market monitoring. For all this, one of the main tools is allergen testing, right? To be sure that the labeling is correct, and of course to do the post-market monitoring. So we could say that there are many emerging allergens, but right now the most that we can or are more visible right now are buckwheat, pea protein, and insect protein. So if we go for buckwheat, so buckwheat is the major food and officially an allergen in Japan and Korea. So even if we mention that the European regulation does not include the buckwheat, it is already included as an allergen in Japan and Korea. And there are also reports of it triggering severe allergic reaction elsewhere, including Europe. So it's only mentioned in the Japan and Korea regulation, but we have there are reported reaction in Europe and in the rest of the world. So buckwheat can be made into flour, but it's not the same family as wheat. So even if we see that it's a very similar name, it has nothing to do with wheat. Despite its name, buckwheat is not a true cereal, and it's not a member of the grass family. So as wheat and buckwheat, they are not related. So usually people that are allergic or a celiac or have some kind of reaction to gluten, they usually can eat buckwheat. In a way that currently buckwheat is a very common use substitute of the gluten in the gluten-free product or to follow a gluten-free diet. It means that more and more we can see buckwheat as an ingredient when we go to buy different products in the supermarket. And so now we need to take care not only for people which is allergic to gluten, but also for people which is allergic to buckwheat, because it's more and more present in the market. So the kit we have currently available from Gold Standard Diagnostics for detecting buckwheat is a high sensitive detection system and is particularly capable of the quantification of buckwheat in grain flours, cookies, pasta, and meat. It is validated in food, swab, and wastewater samples. So the characteristic of the kit is very similar to all the rest of the kits that we have in our ELISA portfolio. So the extraction procedure is exactly the same as the other ELISA. It means that, as I have already mentioned before, the same extract can be used for the analysis of buckwheat and also for the analysis of other allergens. It's really, really convenient because once you do an extract, you can extract a big amount of sample, and then with the same extract, you can procedure with different, almost all kind of ELISA kits. We also have the 48 and 96 wells format, and here you can see the main features. So 2 ppm of buckwheat. In our kits, I would like to mention that this 2 ppm is related to the commodity itself, but we also offer the possibility to express the results in ppm of the protein by using the conversion factor from the USDA. The recovery, this is range between 78 and 101%. It depends on the type of matrix. So when we do the validation of the kit, we test recovery in different kind of matrices, and of course some of them are more complicated, but the recovery is usually quite well. And the incubation time is exactly the same, 20 plus 20 plus 20 minutes. So the characteristics is similar to the rest of our ELISA kits. And I think it's one of the first one from the emerging allergens, because at this moment, I think that probably the technology or the industry needs to go more quickly than the regulation, and we need to have this kind of tools to prevent any kind of allergic reaction for this kind of emerging allergen, even if they are not in the regulation yet. So with this, we have seen all the different kits or the different options that we have in GSD. We have seen the updated regulation regarding allergens. The regulation in allergens usually goes quite slowly, but the VITAL program is more active. The Allergen Bureau is quite aware about all the needs of the patient and the allergic people, and this is why they already have the version 4.0, because they update this reference those values considering the last research investigation. So as a conclusion, I can say that an adequate control and allergen management is one of the current challenges for the food industry. So for this, GSD offers all possible solutions and support to approach this challenge from different angles, so and different techniques. We are seeing manual, automatic, different ELISA, LFD, PCR that we have not mentioned today, but we also have a portfolio for PCR kits, and with all meeting the allergen regulation requirements that are already in Europe and also in some other countries. So with this, I can say to all of you, thank you very much for listening. And now we go to the question part. As Gabor has mentioned, please feel free to write all your doubts, questions, or wherever in the chat. And now we will proceed to, Gabor will proceed to assess them, and we will try to answer them as good as possible. Thank you very much.
Thank you very much for your attention. See you next time, and have a nice rest of the day. Goodbye.
Thank you so much, and goodbye. Thank you.